Hello, my name is Amander Clark, and I am Professor and Chair of Molecular Cell and Developmental Biology at UCLA. I'm also a member of the Eli and Edith Broad Center for Regenerative Medicine and Stem Cell Research, also at UCLA. It's my pleasure to be here today representing ASRM, and I'm very happy to be invited to present this talk, which is New Sperm Sources, Is It Possible? So I have no disclosures. As we all appreciate at this meeting, infertility is a significant problem in today's society. In fact, 1 in 8 couples have difficulty getting pregnant and carrying a baby to term. So infertility, as we know, is treated using assisted reproductive technologies. And there's a whole suite of different technology that can be used. And I'm showing 2 major ones here on this slide. One is using IUI, which involves inserting sperm into the uterus so that it is closer to fertilizing the egg. Another technology for assisted reproduction is in vitro fertilization, which is fertilizing the egg outside of the body with the sperm in order to create embryos that are transferred in order to establish a pregnancy and to have a baby. But these assisted reproductive technologies today require gametes. And so what this means is each couple that is entering into family building needs to make a gamete that is fertilization competent and capable of supporting development in order to make a baby. So what that means is being able to make a sperm cell or an egg cell. And that is because these are the only 2 cell types in the body that enable you to become biological parents. But what about if a person has no gametes? And there's lots of different circumstances under which this can happen. For example, certain medical treatments can damage the gametes such that the gametes can no longer be produced. And good examples of this, for example, are chemotherapy and radiation therapy, which is why so many survivors of cancer following these types of cancer treatments are at risk of infertility because of damage to the gametes. Individuals who have had their pelvic region damaged as a consequence of, for example, war and their ovaries or testes destroyed, this also will destroy the gametes as well. Individuals who undergo surgery where the testes or ovaries are removed, this also leads to loss of gametes, and a good example of this is with transgender surgery. And finally, some people just don't make gametes anymore, either because they're too old or because they never made gametes in the first place. And we understand very little about this category of patient because we don't understand the majority of genes that are involved in making sperm inside of the body. And so therefore understanding defects that are associated with spermatogenesis. So currently, there is no therapy in order to help a patient who has been diagnosed with infertility and is found to have no gametes. There is no way they can have a biological child of their own using the technology of IVF, because of course that requires beginning with gametes, a sperm or an egg. A new technology that's currently being developed in the lab is the technology of in vitro gametogenesis. And what in vitro gametogenesis involves is starting with cells, either 46XX cells or 46XY cells. These are diploid cells, differentiating them in the lab into a gamete. In this particular example here, I'm showing a sperm cell. These sperm cells could then be used to fertilize an egg to create an embryos, and these embryos could be used for transfer. So in vitro gametogenesis is a technology that would precede the use of IVF in order to enable an infertile person who makes no gametes to be able to have a baby of their own. So the question you might be asking yourself is what cell types, what diploid cell types could you begin with in order to differentiate gametes in the lab through the process of in vitro gametogenesis? So one excellent source of starting cells are in fact found within the testes themselves. So it in theory could be possible to perform in vitro gametogenesis starting from less mature, in this particular case, male germline cell progenitors. So for example, starting with spermatogonia, these could be coaxed to differentiate into spermatocytes and ultimately into sperm. So in fact, this technology has been developed using organ culture of the mouse testis, and using these organ cultures grown in the lab, it has been shown that spermatogonia can be differentiated all the way into sperm that are fertilization competent and able to support embryo development. But what about for those individuals that don't have germline progenitors in the testis? So they don't have any spermatogonia anymore. These individuals have, for example, Sertoli cell-only syndrome diagnoses. Well, then there aren't any progenitors in the testis that you could use to differentiate sperm. And in fact, there's no other cell type in the body that can be used to differentiate sperm in vivo, because we know that the germline, the cells that are responsible for making the progenitor cells in the testis and in the ovary for that matter as well, these germline cells are set aside way back in prenatal life as the embryo is implanting into the uterus and lineage specification to create ectoderm, mesoderm, and endoderm is being established. That's when the germline is set aside as well. And these early germline cells are called primordial germ cells. Once these primordial germ cells are made, no other cell type in the body is capable of differentiating all the way into these gametes. So then, how can we make primordial germ cells? So one approach is to begin by making human-induced pluripotent stem cells. So what this technology involves is starting with a cell that comes from your infertile patient. It could be a skin punch biopsy, or it could be a blood draw and isolating the PBMCs, taking those somatic cells, which on their own will never differentiate into gametes, but by using molecular biology, you can turn this fully differentiated somatic cell into a stem cell type called a human-induced pluripotent stem cell. These human-induced pluripotent stem cells can be used to differentiate any cell type in the body in order to restore cell lineages that are damaged or diseased due to damaged or diseased in the body. And in this particular example, the human iPS cells could be used to make gametes. So the proof of concept that human iPS cells could be used to make gametes, in fact, comes from research using the mouse. So in these particular experiments, a little biopsy is removed from the tip of the tail of the mouse. Fibroblasts are grown out of these biopsies in a process known as derivation. These fibroblasts are reprogrammed into induced pluripotent stem cells, and these induced pluripotent stem cells can be used to differentiate a cell type in the lab called a primordial germ cell-like cell, or a PGC-LC. iPS cells have also been used to transdifferentiate directly into oocyte-like cells. However, these cell types are not fertilization competent and do not support embryo development. So I'm only going to talk about the differentiation of primordial germ cell-like cells today. Primordial germ cell-like cells, or PGCLCs, as you'll hear me refer them to, are equivalent to the primordial germ cells that are found naturally inside of the body during early embryo development. What distinguishes them is the like cell nomenclature, and this is to show that these cells are differentiated from pluripotent stem cells, either induced pluripotent stem cells that, say, could come from a tail-tip fibroblast, or embryonic stem cells that can come from embryos that are left over from the IVF clinic. And so these primordial germ cell-like cells are equivalent to that fully specified germline progenitor, which is competent to ultimately differentiate into the gametes. Using these primordial germ cell-like cells, fertility has been restored in infertile mice. And I'm going to go through these experiments today. I want to say from the outset, these pioneering experiments have been performed primarily by Katsuhiko Hayashi and Mitanori Saito in Japan, and now they're being replicated around the world. What this involves is beginning with primordial germ cell-like cells. In one of the first studies, this was in 2011, so a decade ago now, these primordial germ cell-like cells were transplanted into the seminiferous tubules of a newborn mouse. And in that relatively immature niche, these relatively immature primordial germ cell-like cells were capable of maturing, forming sperm, and the sperm could be used to fertilize an egg to give rise to embryos and ultimately offspring. In an innovation that was published in 2016 by Mitanori Saito's lab, these primordial germ cell-like cells were differentiated further in the lab to form germ cell-like cells, which are similar to spermatogonia. Then these spermatogonia can be transplanted into infertile adult male recipients, and under these circumstances, the germline stem cell-like cells will differentiate into sperm that can be used to fertilize an egg to give rise to embryos and offspring. This technology is not only available to infertile males, but also to infertile females. And in fact, female primordial germ cell-like cells have been transplanted into the ovary in order to generate oocytes that could be fertilized in order to generate embryos and offspring. And even more excitingly, this technology of organoids has been used to make gametes. And that involves starting with female primordial germ cell-like cells, combining them with ovarian somatic cells to make organoids that entirely in the lab will self-assemble and then give rise to eggs from those primordial germ cell-like cells that came from the iPS cells that originally came from the skin fibroblast. So in humans, it's very reproducible and reliable to make human primordial germ cell-like cells from human pluripotent stem cells. And what this involves, one of the approaches, the one that we've been using in our lab for many years, is starting with human-induced pluripotent stem cells, differentiating them into a precursor intermediate known as an incipient mesoderm-like cell, and then differentiating that in three-dimensional aggregates in individual wells of a 96-well plate into a mixture of embryonic cells that contain somatic cells and PGC-like cells. So if we look inside one of these aggregates, which is found in one of those wells in a 96-well plate, this is what it looks like. And in blue are all the different nuclei of the somatic cells within the aggregate. And in color are the primordial germ cell-like cells that are forming within a niche within the aggregate. So over the last number of years, my laboratory has been performing a lot of genomic studies to determine if the primordial germ cell-like cells we make in the lab are similar to human primordial germ cells that are found naturally inside the body. What this data shows is when we perform RNA sequencing of these germ cells, we can see that these primordial germ cell-like cells, which are shown here, are clustering on the same axis as human primordial germ cells and are distinct from human ES cells and that intermediate human-induced, incipient mesoderm-like cells. This is a different way of displaying the data. This is the germ cells shown on this side. This is the human ES cells and IMES cells shown over here. So we now appreciate transcriptionally that human PGC-LCs are similar to human PGCs found in the embryonic body. So the next step is, how do we get them to differentiate? So this is some beautiful work that was recently published by Kotaro Sasaki at UPenn and colleagues. And what he did was to develop a technology called reconstituted testes. What that involves is starting with mouse embryonic testes, depleting the germ cells, the primordial germ cells from the testes, and then taking that single cell suspension of cells and aggregating them for 2 days in this reaggregation well, which is a floating culture, which causes them to form a little aggregate. Those aggregates are then transferred to an organ culture membrane where they can be cultured. And this particular example shows you those organ cultures after 14 days. And what you can appreciate is that single suspension of cells self-assembles into an immature testicular-like structure with seminiferous tubules. So now, using the reconstituted testis, a niche can be created for differentiating the PGC-like cells towards sperm. So the next step that his lab did was to take the human iPS cells, differentiate them into incipient mesoderm-like cells and then into human primordial germ cell-like cells, and combine them with that single cell suspension of mouse testes that had been depleted of endogenous primordial germ cells. And the first step is to create that floating culture. This is called a xenogenic reconstituted testis, and it's xenogenic because the somatic cells originate from the mouse embryo, and that's shown in pink, whereas the germ cells originate from the iPS cells, and they're shown here in green. When you culture these organ cultures for up to 120 days, we can see that the human cells, which are marked here in red by TFAP2C, they persist within the seminiferous tubules of the testis. Not only do they persist, they also differentiate into immature spermatogonia, as shown by markers of immature spermatogonia, such as MAGE-A3 and MAGE-C2. And in this FACS plot, where the iPS cells have a reporter for a prospermatogonial marker, PB-like4, we can see that between 80 and 120 days, these primordial germ cell-like cells will start to differentiate and express markers that are found in spermatogonia. So this is a really important observation because Brad Cairns, Jingtao Guo, and I showed that fetal germ cells, the prospermatogonia that are found within the fetal testis, which are the ones equivalent that are made in these xenogenic reconstituted testes, are very similar to state 0 spermatogonia that are found in adults. And so this is a single-cell RNA-seq study showing the progression of germline development from early embryonic to fetal stage to these PB-like4 fetal prospermatogonia, which are clustering together with the adult spermatogonia before they differentiate through meiosis. Given this promising result using human cells, my lab has moved forward with developing preclinical models for differentiating iPS cells in the rhesus as a proof of concept that sperm can be made in primates, of which human is a primate. So we use the rhesus macaque, and these studies are performed in very close collaboration with the Oregon National Primate Research Center and John Hennebold, as well as Kyle Orwig at the University of Pittsburgh. So what we do is generate rhesus-induced pluripotent stem cells, differentiate them the exact same way we would with human cells into aggregates, and we can show using FACS that primordial germ cells emerge within these aggregates starting at around day 2 to 4 of differentiation. When we transplant these aggregates made in rhesus into an adult rhesus testis, what we can show is that these germ cells will start to mature within the testis and form cells that have some hallmarks of prospermatogonia. So in summary, what I've told you is that assisted reproduction and IVF today is only possible if your patient produces germ cells and gametes. What we think is that iPS technology could potentially be a cell-based therapy to restore the ability to make gametes. In the cases I've shown you today, it's sperm. This is through the technology of in vitro gametogenesis, or IVG. IVG can currently generate fetal stage spermatogonia, but what we appreciate is that fetal stage spermatogonia are transcriptionally similar to adult spermatogonia. Differentiation into sperm has not been shown yet, but it'll only be a matter of time. So what this means is that international standards and guidelines are now required to create a framework for testing sperm quality from IVG. And this involves a framework for using embryos for research purposes to test fertilization potential of IVG-derived gametes, and also developing the ethical and safety standards for one day potentially translating this technology to humans. So I'd like to thank you very much. I'm representing the ASRM at this conference, Reproduction Reimagined, and I'd like to thank you very much for your attention.